Hello, everyone, and welcome to today's EBD Group webcast titled Lab in the Loop Hit Discovery, Unlocking Undruggable Targets with AI-Powered Hit Identification Platform, sponsored by Axcelead. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. And in 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our speakers for the session. We have Akito Hata. He's the head of Screening Business Unit and Digital Unit for Axcelead Drug Discovery Partners. And we have Hiroshi Kajino, Principal AI Scientist for the Digital Unit of Axcelead Drug Discovery Partners Incorporated. Thank you both for joining us today. I'll hand it over to Akito to begin the presentation.
Yeah, thank you very much, Mariana, for introduction. And thank you very much for everyone joining the webinar. My name is Akito Hata, and I'm the head of Screen Business Unit and Digital Unit at Axcelead. I have been working with in vitro biology and screening field more than 15 years. Prior to joining Axcelead, I had worked at Takeda in both Japan and Boston site. Hiroshi, do you wanna?
Hi, I'm Hiroshi Kajino, and I am a principal AI scientist for Axcelead Drug Discovery Partners. And I have been working on machine learning for more than 10 years since I graduated the University of Tokyo and obtained a PhD degree from the university. After getting a PhD degree, I joined IBM Research Tokyo and have spent 10 years to develop basic foundational machine learning techniques, as well as some applications of machine learning to several business clients. Thank you.
Yep. So since this is the first session in our webinar series, I'd like to begin with a brief introduction to our company. In the upcoming webinars, we plan to cover topics such as medicinal chemistry, translational research, and in vivo models. So please stay tuned for those as well. So Axcelead is Japan-based integrated drug discovery solution provider located in Greater Tokyo area. Shonan site, Axcelead Drug Discovery Partners was established in 2017 as a spin-off company from Takeda Pharmaceuticals. Since 2019, Axcelead is fully owned under the fund managed by WIS Partners, and we are fully independent from Takeda. And Shino site, Axcelead Tokyo West Partners was established as a joint venture company with Teijin Pharma in April 2024. The unique point of Axcelead as a drug discovery CRO is that we have inherited assets from Takeda and Teijin. For example, most of our 300 researchers have extensive experience in drug discovery projects at Teijin or Takeda. Second, we have inherited pharma-grade research facilities and assets, including 1.2 million original compound library. And finally, we have access to the project data and information with more than 1,000 projects, which are from Takeda and Teijin. Leveraging these unique features, we work closely with our clients to provide integrated drug discovery services or platform services. As for the business truck vehicles, we currently have more than 290 clients from all over the world, and we have more than 90% of repeat rate. Recently, we signed collaboration agreement with Lilly for multiple drug discovery programs and master service agreement with Acadia to support developing novel medicines. So we are expanding our reach to the US, Europe, and other sides of the world as well. And Axcelead has a comprehensive drug discovery research functions required for preclinical drug discovery. And we can provide a full range of services from target ID, screening, lead optimization through IND. We have extensive knowledge and experience in small molecule drug discovery. In addition to these core strengths, we have built strong capabilities in emerging small molecule modalities, such as targeted protein degradation and RNA targeting small molecules, as well as in other therapeutic modalities, including peptide and nucleic acids. More recently, we are also expanding our capabilities in the field of antibody drug conjugates as well. Leveraging a robust network built through strategic partnership, we contribute to our clients' drug discovery projects. So in today's webinar, we will be talking about our hit identification and AI capabilities to accelerate drug discovery, focusing on our solution against difficult targets. First, I'm going to introduce our HTS platform. After a therapeutic target has been selected, the next step is to generate lead compounds that enable efficient lead optimization stage. Beyond showing robust activity against the target, lead compounds must satisfy multiple criteria. For example, a structure activity relationship information rich enough to guide optimization strategies, favorable ADME profiles, acceptable toxicity risk, and clear paths to securing intellectual properties, and so on. Improving the probability of downstream success starts from hit identification stage. Discovering variety of high quality hit compounds directly increases the odds of delivering a viable lead candidates. For especially difficult targets, common causes of failure include low quality assays that yield a false positive or overlook weak but meaningful activity, assays with a limited disease relevance, and most critically, screening library that are small, lack chemical diversity, or are poor in drug likeness or novelty. In the hit to lead stage, a failure often stems from an insufficient portfolio of chemotypes. When promising series are lost because SAR insights are shallow or ADMET profiles are unfavorable, the absence of enough backup chemotypes leads to program termination or significant delays. So at Axcelead, we have established a platform that overcomes these challenges and reliably generates lead compounds even for difficult targets. In hit identification stage, leveraging a proprietary high-quality compound library and decades of screening know-how, we deliver high-quality hit series with an exceptional success rate. When this foundation is combined with our AI-driven hit expansion engine, we not only accelerate initial hit expansion from our physical library, but also mine virtual chemical space restricted to compounds which are with favorable ADME properties. The result is next generation lead generation package that balances potency, developability, and novelty in very early stage of the project. In this webinar, we will introduce our hit finding and AI solutions, including a case study for a difficult target. First, I will talk about the hit identification platform. At Axcelead, we offer deep screening expertise built on foundation of our 700 HTS campaign experience, including the one inherited from Takeda. Since Axcelead started business, we have successfully identified hit compounds in over 98% of all clients' projects. The chart here illustrates the distribution of target classes across these campaigns, including many typically considered difficult targets, such as class B GPCR, ion channel, protein-protein interaction, and RNA binders, and so on. Yet we have consistently maintained exceptionally high success rate in hit finding. We have received high positive feedback from our clients For example, in one case where no tractable compounds were found using client's library, our screening led to the identification of hits that successfully progressed into lead optimization. In another case, a client was surprised by the unexpectedly high number of high-quality hits discovered for a challenging target. Our performance is driven by two key features. One is the strategically tailored screening workflow defined through 700 HDS experience. The other one is original 1.2 million compound library inherited from Takeda and Teiji, which provide a chemical diversity, drug likeness, and novelty. Together, these assets enable us to uncover high-value hits quickly, even against the difficult targets, and give our partners a good starting point for following stages. In this slide, we present our proven track record in assay development, which is essential for successful hit identification. Leveraging our extensive HTS experience and expertise, we can propose optimal assay systems tailored to each project in close consultation with our clients. Over the past 6 years, we have established more than 480 assay systems with over 80% developed in-house at Axcelead. Assays can also be transferred from client as well. So we have received high positive feedback from clients regarding our assay development capabilities as well. For example, in some projects where assay development was expected to be challenging, we were able to complete it faster than anticipated. In other cases, our assay design proposal enabled high-quality HTS to be conducted effectively. And a client have also commented that the data quality we deliver is superior compared to other CROs. With this solid foundation, our assay system are seamlessly integrated into reliable and high-quality HTS platform. This slide shows overview of our proprietary compound library. 1.2 million compounds were inherited from Takeda and Teiji, and they are stored in our fully automated warehouse located in Shonan area, the same location we conduct HTS, which enables quick compound sourcing. 60% of compounds were synthesized in-house. That means they are likely to be commercially not available original compounds. Therefore, there is a high potential to deliver hit compounds with structural novelty. In addition, our compound library maintains a high average drug likeness score of 0.62, enabling the identification of hit compounds that are most likely to be suitable for further optimization. For the random screening, we have single diversity library consists of 417,000 compounds and pool library of 500,000 compounds. Pool library means 10 compounds are mixed in one well to maximize efficiency of primary screening. We also have variety of focus libraries, target-oriented, chemical property-oriented, covalent annotation, and so on. And based on our HTS experience, we propose the strategy to select combination of libraries to be screened for the target. We also have combinatorial virtual library of 10 billion compounds. In combination of more than 80,000 original available building blocks and high-throughput chemistry platform, this enables efficient SR analysis, particularly in early stage of lead generation. So far, I have introduced our HTS platform, which has consistently delivered high success rate across various projects, including difficult target. So now I'll hand it over to Hiroshi, who will walk you through our AI solutions for the hit expansion stage.
Okay, so from now on, I'm going to explain about our AI capabilities as well as our AI-powered hit expansion system. First of all, I'm going to give a brief overview of our AI assets. The first item is our proprietary data, which are inherited from Takeda and Teijin, and which have been consistently accumulated for more than 25 years. The data consists of two parts. The first part is unmet tox data, which includes more than 20 in vitro and in vivo endpoints. And for each endpoint, we have this amount of examples. And the other side of the data are activity data, which is targeted to around 1,000 of unique targets. And we have these numbers of data points for each of the targets. The second item is a predictive model. So we have such a proprietary data, and given such a data, we can build multiple predictive models for unmet tox prediction as well as activity prediction. And we take the foundation model approach, which is a very modern machine learning approach to train a good predictive model. The approach consists of two steps. The first step is to train a foundation model using all of the data that we have. And the second step is to fine-tune the foundation model into individual targets. And by using these two steps training, we can build a good predictive model for each of the endpoints. And we show some of the performance metrics of our predictive models. In the graph shown in the bottom, we show the performance of our unmet tox predictive model, which is shown in the green bar, and the performance of a commercial software, which is shown in a blue bar. And as we show in the graph, in all of the endpoints, our predictive models are better than the commercial software in terms of Pearson correlation metrics. In addition to the unmet tox prediction model, we have also built several activity prediction model using our data. And for most of the targets, our predictive model has the R square of our prediction model is around 0.4 to 0.5, which is very good performance for virtual screening as well as lead optimization. And the final item is a generative model. So we have several types of generative model, including de novo generative model, scaffold constraint generative model, and the reaction-based generative model. And all of these generative models are trained on our own chemical space so that the generated structures are easy for our medicinal chemists to synthesize the compounds. And for especially for reaction-based generative model, this model uses a synthetic pathway to generate a structure by using our own building blocks and our chemical reaction templates. And that results in more than 10 billion possible products. And given such a reaction-based generative model, the good point of this model is that we can actually synthesize the generated structure by our parallel synthesis platform, whose success rate is around 90%. And also the throughput is around 100 to 200 compounds per week. So from now on, I'm going to explain how to combine our AI capabilities with HTS so as to lead to our next generation lead generation platform. And we have two types of scenarios. The first scenario is hit expansion. And in this case, we first have to run HTS campaign, and we have assume we have tens of hit compounds from HTS campaign. And given such a result, the first step is to train an activity prediction model by using the outcome. And then by using this predictive model, we run additional virtual screening of our physical library to get a virtual hits out of our 1.2 million physical library. And then given such virtual hits, we run another round of HTS on virtual hits to confirm whether virtual hits are actual hits or not. And by using this kind of workflow, we can discover novel hit compounds that have never been accessible by the first HTS. And preferably, we can find novel hit compounds which are dissimilar to existing hits. And the second scenario is more aggressive scenario where we use a virtual library instead of physical library. So by using a virtual library, which is a billion-sized library, we can explore a vast chemical space that has never been possible without AI capability. So by using this virtual library as a source of virtual screening, we can obtain many more virtual hits out of this virtual library. And then given such a virtual hit, we can run parallel synthesis to actually synthesize the compounds, and then run HTS again to confirm whether the virtual hits are actual hits or not. By using such a virtual library, we can discover novel hits which have never been accessible with the previous HTS platform. And from now on, I'm going to explain a case study of scenario 1, whose target is a class B GPCR antagonist, where only antibodies are currently available under clinical stage. So in this case study, assume that we have a first screening outcome, which consists of 180,000 examples on target inhibition percent at 10 micromolar. And also we have 640 examples on dose response curves. And by using such data for training activity prediction model, we can get a good prediction model here. And also we can run a virtual screening by using this predictive model. And from now on, I'm going to go into the details of each of the steps of this workflow. So first of all, I'm going to explain the performance of our predictive model. So as explained in the previous slide, our predictive model is based on a foundation model, which has been trained by our own all of our proprietary data. And the test scores of the inhibition percent at 10 micromolar are as shown in the upper side of the slide. And in particular, if we focus on the coefficient of determination, R squared score on the test set, the score was 0.55, which is quite good for activity prediction model. And more importantly, this score is much better than the score of the model that is trained without our foundation model. So this performance difference shows that that our foundation model approach is quite useful for building a predictive model for activity prediction. And also, if we focus on a binary prediction where the predicted value is whether or not the compound is active or not, the precision score is around 0.7, which says that out of 10 virtual hits, 7 compounds will inhibit the target for 50%, which is a good number for activity prediction models. Also, the recall score was better than that without foundation models. Overall, these show that our predictive model performs better than the standard one, especially thanks to our foundation models. In the next slide, I'm going to explain how to run virtual screening using our predictive models. Here is the experimental protocol for the virtual screening. So first, we select compounds from Axcelead physical library that are not tested yet for this and that will not be tested by similarity search, and that are in stock and that have highest predicted activities on the target. And from such compounds list, we selected top 1,020 compounds for actually assay. And we tested the inhibition rate of these compounds in the actual assay. And the graph of the figure on the right-hand side shows the similarity distribution of the compounds selected by our AI-based hit expansion and similarity-based hit expansion, which is a traditional approach to expand hit compounds. And the x-axis shows the maximum similarity to the existing hit compounds. So as we go in the right-hand side, that shows that the compound is more similar to the known compounds. And this histogram shows that our AI-based hit expansion can select not only those similar compounds, similar to the existing hit compounds, but also compounds that are not similar to the existing compounds. This is in contrast to the traditional similarity-based hit expansion, because the traditional approach can select mostly from those compounds that are similar to the existing hit compounds. And the third step is to physically evaluate the activities of virtual hits by HTS. And this table shows the statistics from the actual assay outcomes, and each row shows the result for each definition of hit. For example, in the first row of the table, the definition of hit is that the inhibition to the target at 6 micromolar is more than 30%. And given this definition of hit, the similarity-based hit expansion hit rate of this approach was 33%. In contrast, for the AI hit expansion system, the hit rate of our system was 82%, which was much better than the similarity-based approach. And as we change the definition of hit compounds, we show the same trend, showing that our AI-based hit expansion has much higher hit rate as compared to the hit rate of the similarity-based hit expansion. This is one advantage of using AI-based hit expansion. And more importantly, the other advantage of using AI-based hit expansion is that our platform can find those molecules that are not similar to the existing hit compounds. In the right column of the table, we show that we have 275 compounds that are not similar to the existing hit compounds. And out of these virtual hits, if we set the definition of the hit to be inhibition rate to be more than 30%, then the hit rate was 64%, which is still much better than the similarity-based hit expansion. And the most important thing is that these hit compounds would not be accessible without our AI-based hit expansion system. So this is a very unique feature of our hit expansion system. And finally, we will give a brief overview of the dose response curve and a Q log AC50 metric. So these graphs shows that out of the hit compounds that are out of hit compounds coming from the third step of our protocol, we compare the statistics of the hit compounds we have obtained from similarity-based search and AI-based hit expansion. And this shows that our AI-based hit expansion has much better Q log AC50 scores as compared to those molecules discovered by the similarity-based search. And also, if we compare the selectiveness of the compounds, our AI-based hit expansion still has much better scores as compared to the similarity-based hit expansion. And in conclusion, as we have seen in these actual assay outcomes, our AI-based hit expansion has much better capability than the similarity-based search, which is a unique capability of our AI integrated HTS platform.
Okay, thank you, Hiroshi. We hope this webinar has provided a clear overview of how our integrated HTS platform and AI solutions can accelerate your early drug discovery, including difficult targets. We are looking forward to working together and supporting your next project for HTS and early lead discovery. Thank you very much for your time and attention today. And now I will hand it over to Brianna to take questions from audience. Thank you.
All right, thank you, Akito and Hiroshi, for a wonderful presentation so far. Before we begin today's Q&A, let's run through some brief announcements. First, I would like to thank Axcelead very much for sponsoring today's event. We have a few questions already in the queue, but if you have a question for Akito or Hiroshi, please submit it now into the Q&A module on the left-hand side of your screen. All right, with that, Akito, let's start with the questions. Hiroshi, Akito. Our first question today. In your presentation, you highlighted a case involving a Class B GPCR. I'd like to ask whether the hit finding capabilities and AI technologies you've described can also be effectively applied to other types of difficult targets, such as protein-protein interactions or RNA binders.
Okay, so as we introduced in the presentation, the success rate of hit finding is 98%. That is including difficult targets like PPI and RNA binders. Along with high quality diversity library, we also have focus library for specific target classes like GPCR, molecular glue, and RNA targeting small molecules. Especially for RNA binders, we have internally synthesized splicing focus library and other general RNA targeting library as well. And recently, we have feedback from a client that our library resulted in the highest hit rate ever. So by carefully choosing appropriate library for each target, we can deliver successful hit finding for difficult targets, including Class B GPCR and other target classes as well. So in terms of AI capability, Hiroshi, could you add some comments?
Okay, so short answer is we can apply our method to any target class. And this is because our method is a ligand-based approach, and that is target agnostic, meaning that we don't need any crystal structure of the target. Thank you.
All right, thank you both. Our next question today. There are other companies that offer software for ADME and activity prediction using AI models. In comparison with those tools, what would you say are the key strengths or differentiators of your approach?
So maybe I can answer the question. A key differentiator is the dataset to train our ultimate tox prediction model. Many other companies are using public data to train the models, but the sample sizes of the public data are relatively low. And also, we have to do careful manual curation to get high quality data. In contrast, our models are trained on tens of thousands of data points for each endpoint, which are obtained by consistent experimental protocols, which leads to the high predictive performance as compared to the other models provided by other parties.
All right, very good. Our next question here. They say, if I understood the case study correctly, the AI model was built using data obtained from high throughput screening. Do you see any potential to reduce costs by using virtual screening for hit identification instead of HTS?
Okay, so I can answer the question. So the platform that we have explained have used HTS as the first step, but we can exclude this step if we have plenty data for the target. So we have inherited data from Takeda and Teijin, and which includes target activity data. So if the target of interest is included in our database, then we can skip the HTS campaign here, and we can start building a predictive model from our data, which are inherited from Takeda and Teijin.
All right, very good. Another question here. I'm very interested in your original HTS library. We would like to explore the possibility of screening it using our own assay system. Would it be possible for you to provide the compounds from your library for such a purpose?
I will answer that question. So we don't provide our original HTS library compounds to external screening for screening use at this moment. However, if you established assay system which you would like to conduct HTS, we can have your assay system transferred to Axcelead, and we can perform HTS at our end. And also, if you would like to assess the quality of our HTS library, we can provide fingerprint information to compare our library and your library. So please feel free to reach out to us if you are interested. Thank you.
Thank you, Akito. You presented performance comparison data for your unmet prediction model. What data sets were used and how was the performance evaluated? I'm curious to know whether the comparison conditions might have been set in a way that favors your model.
Okay, so I can answer the question. So we have used our own data for training our model, as well as testing our model and industry standard AI models. So in that sense, since the data set is coming from our chemical space, that could be favorable for our chemical space. But when comparing the performance, we take a very careful approach so as not to be as much as fair as possible. The way we split the data is as follows. So we have our own data set here, and we split the data into training for training a model and for testing the models. And the split was done by a scaffold split, which is a de facto standard data split method to check the performance, to check the predictive performance on unseen chemotypes. And by using such a split, and so by keeping the test set separately from the training set, this performance comparison is as fair as possible.
All right, thank you. And I think we have time for one more question. Could you please give an idea of the typical timelines for both hit identification and subsequent AI-driven hit expansion steps?
Okay, thank you. So the timeline depends on the target and assay system, but typically assay development takes 4 to 8 weeks after materials are in place. And for the small-scale screening, the screening itself can be completed within 2 to 6 weeks. And AI model building, virtual screening, and in vitro assay evaluation against the virtual hits, generally it takes 3 to 4 weeks. So in the best case scenario, the entire process can be completed in 2 to 3 months.
All right, very good. Thank you, Akito and Hiroshi, for an excellent session. And thank you once again for Axcelead for sponsoring today's session. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. Now, this session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And please take a moment to complete the feedback form that should be appearing on your screen so we can improve your digital week experience. On behalf of the EBD group, thank you very much and have a great day.